Hey, you guys. I'm not going to be in class for a good portion of the class tomorrow, so I wanted to give you a video to watch. This should only be, 5 or 6 minutes, so that you can get a head start. So when we get into class, you can just get started on the lab. I'll be there till about 8, and then I have to get going. So we're going to be doing an onion root tip mitosis lab. We are examining onion root tips under the microscope. And we are looking at a slide, a moment in time of an onion root tip. And we're looking at an onion root tip because a root tip is going to be doing lots of mitosis or lots of cell division because it's growing. And so therefore we're going to see lots of stages of growth in each of the different cells. And so this is two different captions or two different moments of time. And we can see all the different stages of mitosis. And it's going to be really important for me to go over the different stages, what they look like, and then what the lab is going to entail. So here, if you remember, mitosis starts with interphase, or interphase precedes mitosis, I should say. And so during interphase, the chromosomes are in those loose, relaxed form called chromatin. And so that loose, relaxed form, it looks like this. Think of it like this, right here, right here. These are going to look like your solid dots. They look like solid dots because if you think of like a ball of string, you can't see each individual string. You see just the whole ball, right? And so this is what you see right here is the ball of string. You can't really identify any of the separate strings because they're just so thin and they're just kind of like a clump, like we said, like kind of like a bowl of spaghetti. They're just sort of laying there. And so these are going to be your cells in interphase, your normal cell life. This should probably be the most abundant phase that you see because cells spend most of their time in interphase. And so as we go into prophase, we know that the chromosomes are going to start to coil up from that chromatin form into chromosomal form. And we're going to be able to start to see them to be defined. And so you can start to see here the definition coming along. You can see definition very well here. You can see this looks like right here. This is, you can see definition starting to form. So it's no longer just a ball of string. It is now those strings starting to coil up, and so you're starting to see that definition. But it isn't. The chromosomes haven't really moved around and made any shapes yet. And so once we start going into metaphase, remember meta, middle. During metaphase, the chromosomes line up along the middle. And so here we can see how the chromosomes are lining up in the middle of the cell. And here we can see that again, chromosomes are lining up in the middle of the cell. This is metaphase. So remember that each stage doesn't just start and stop neatly. They sort of blend into one another. And so let's see if we can find something that looks like it's kind of in between. So this looks like it might be in between here. Do you see how it's in that, it's like really late in that prophase stage and it's kind of starting to look like it's going into that metaphase stage. It looks like those chromosomes are moving towards the middle, really late prophase, whereas this would be really early prophase. And so it's not always super easy to define exactly, this is definitely metaphase. Sometimes it is, like this is very obviously metaphase. And this is very obviously prophase. But you're going to have some that are kind of metaphase-y, prophase-y, metaphase-y, or metaphase-y, anaphase-y. And so prophase, metaphase, anaphase. Remember ana apart, A. And this right here is a great example of anaphase. Anaphase doesn't take very long, and so it's hard to find anaphase. You might only see one or two in your screenshots. And even in here, we see only one anaphase. And so you can see how those, there's a distinct separation between the two sets of genetic material, and they're migrating to the poles. And so this right here is your anaphase. And then telophase. Telophase is when the cell has separate material. Let's see if we can find one. I don't see one here. Let me get a different cell. Okay, so here, this is a telophase. Can you see how this is still one cell right here? But there's two sets of genetic material in that one cell. There's lots of great examples here. Let's go through a couple more examples. This is definitely a metaphase. This is a prophase. Ooh, this is a great anaphase. Another anaphase. So you can start to see prophase. There's a lot of anaphases in this one. And you can see right here how the nuclei are still little. These cells are still little, but there's still that cell plate forming in between the two. That is because this just split. It might still be variant. This is definitely end of, like, beyond telophase. There's a cell plate growing here because these two nuclei are completely separate. So this is definitely just like a new separation. All right, so since we have an understanding of what to look for, let's look at the lab. The lab is here. What we're going to be doing is we're going to be taking 3 screenshots, just like the ones we were looking at. And you're going to count the number of cells in an interphase, in prophase, in metaphase, in anaphase, and telophase. And then you're going to find the percent of the cells in each of these phases. So I completed this here. You're going to be the number of cells. Let's say that if we had counted 92 total cells, how many of those cells are in interphase? Let's just say it was 60. And then 5 in prophase, 3 in meta, 1 in ana, 2 in tele. So if we put 60 over 92, if we divide 60 by 92, that means that our the percent of time that a cell is in interphase is 65.2% of the time. Prophase is, so if we put 5 divided by 92, that means that our cell is in prophase 5.4% of the time. So go to the just go to the 10th spot in, actually, why don't you guys take it to the 100th spot, to the 100th place, just because the percents get pretty small for ana and telophase. And then you're going to find out, you're going to do that 3 times here. So how much time round 1, how much time in each phase round 2, how much time in each round, in each phase round 3. Then you're going to find the averages here, and then calculate the percent of time for those averages. And then we're going to find, multiply that percent. So if it's 65.2% of the time, you're going to multiply 65.2% or 0.652 times 24 or 24 hours in one day. And then make sure that you're not multiplying 5.4, but that you're going 0.054. And then when these numbers get really little, that you are moving the decimal. So here it's 14.4 hours of the day that it's in interphase, and then 0.24 hours of the day in anaphase. So once you're getting below an hour, don't worry about changing it to hours and minutes unless you really want to. That's not really the point. Then we're going to be starting to talk about cancer next class, and so this is our introduction. And so this should be pretty quick. Let me know if you have any questions. I'll be there for the first 10 minutes of class and maybe the last 10 minutes of class. All right, I'll see you guys tomorrow.